of some of the main methods that scientists use to measure binding affinity. So, how strongly two molecules bind to one another. Commonly, this is given in terms of the KD or the dissociation constant, which is the concentration of one molecule at which half of its binding partner is bound. Measuring this KD is really important because, well, just because two molecules can bind one another doesn't mean that they actually will. This is all going to depend on their concentrations and their affinity for one another. So let's talk about how we can measure that.
This is not a comprehensive overview because that's not my goal. My goal isn't for you to know the details of each of these different techniques or even to be able to remember all of these names. But it's a good idea to know what's out there, to know the types of techniques that scientists can use to measure binding affinity and be able to recognize them when they show up in papers and not have the first time you be encountered with them be like in the wild like that. So for my class, you're going to be assigned one of these techniques, and you'll have to look in a little more detail and then share with the class and show us an example of how it's used in a paper. But still, that's just really going to be touching the surface, and this is just to expose you to some of these different techniques. So in this video, I'm just going to briefly introduce them, and these are the main ones I'm going to focus on, but there are going to be others as well. Now, some of these methods are going to study just the affinity, whereas others will give you the kinetics as well. So remember that for any reversible interaction, so this could be like a reaction, or this could just be a binding interaction, you can write an equilibrium constant. And one of these constants that we can write is the dissociation constant, or KD, which is the equilibrium constant for unbinding, and much more on that in the video from the other day. Remember that we can define KD in a couple of different ways. So one is in terms of the equilibrium concentrations. So you mix A and B, you let them reach equilibrium, and then you go and look to see how much is bound. The KD is going to be equal to the concentration of free A times the concentration of free B over the concentration of the complex AB. That is, you have the concentration of your unbound over the concentration of your bound. Now, it works out that the KD is going to equal the concentration of one of the partners at which half of the others is bound. So if you can go and you can measure this, you can determine the KD. The weaker binders are going to have higher KDs, meaning they require a higher concentration of A in order for half of B to be bound. So we can get the KD just by measuring the relative concentration of bound and unbound at a range of concentrations of the other partner. But if we do that, what we're losing is the kinetic information. So the KD itself does not tell us about the kinetics. So kinetics has to do with like speeds. And when we're measuring just those concentrations after they've reached equilibrium, we don't know how they got there. An equilibrium could be reached, because of some various combination of K on, so the forward rate constant, and K off, the reverse rate constant. So how quickly do they bind, and then how quickly do they unbind? The more they bind, basically the more you'll find in the bound form, but the quicker they unbind, the less you'll find in the bound form. And so the combination of these two is going to influence how much you find at equilibrium. More specifically, we can say that the KD is equal to our K off over our K on. And remember that these are going to be our rate, that those little k's are our rate constants, and the big K is our equilibrium constant. So we can define KD in terms of K off and K on. If we know two of these values, we can figure out the third. And so if we measure K off and K on, we can find the KD that way as well. But we need to use different methods in order to study kinetics versus equilibrium. Some of the methods we use will be able to give us both, whereas others will only be able to measure the equilibrium constants. So let's get into a few of these methods. Often we refer to these as biophysical methods. And as that name might suggest, they do involve some math, but thankfully there are computers that do that math for us, and we don't really have to worry about it too much. Although it is important to pay attention to the concentrations that we use. I mean, I'll post to some resources to help people set up experiments, but you don't need to worry about those technical details for the context of our class. So these are the main methods that we're going to go over, as well as a few others. And again, don't worry about memorizing all of these. These are surface plasmon resonance, SPR, biolayer interferometry, BLI, isothermal titration calorimetry, ITC, microscale thermophoresis, or MST, fluorescence polarization, FP, slot blots and dot blots, EMSA, Electrophoretic mobility shift assay, and ELISA, enzyme-linked immunosorbent assay. Yes, 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 there are a lot of acronyms in biochemistry, but that's because there's a lot of these words pushed together, and better to have a bunch of acronyms than a bunch of really long phrases. Okay, so let's start with surface plasmon resonance. This is one of those ones that's really good for telling you the binding kinetics as well as the KD. How it works is basically you have this chip, and on the surface of the chip, you have the ligand conjugated to the chip, or you have one binding partner conjugated to the chip. And when I say conjugated, that just means that it's kind of like covalently attached. So it's tightly stuck onto that chip. Now what's going to happen is you have a microfluidic system that's basically going to flow a liquid over the chip that contains the other binding partner. And so basically, we can call this like the analyte often. So you have like the ligand that's on the chip, and then you have the analyte that you're flowing over. Basically, one of those is going to be your A, one of them is going to be the B. And typically, you choose the one that's bound based on how you're binding it. So it could be something that's tagged, and then you have the corresponding thing on the other side. So you might have a streptavidin chip and a biotin labeled tag. You might have some sort of antibody that's going to bind to your thing. You just need a way to attach your compound to the chip, and then you'll flow the other partner over it. Now, why we do this is because when things bind to one another on the surface of the chip, they're going to change the properties of that surface. And one of the properties that they're going to change is basically how the chip is going to refract or reflect light. So basically what's going to happen is there's a light source under the chip, and it's going to shine light at this up from the bottom. This light is then going to get reflected off the surface of the chip, and how much it is reflected is going to depend on what's happening on that surface. If you have a molecule bound, well, now you have this bigger, bulkier thing near the surface of the chip. It's going to change the properties. You're going to get a different angle of reflection, and this can then be measured by a detector. So in this way, you're able to measure the binding, the unbinding, and how much is bound. Now, sometimes you might see one of those curves like what we've been looking at. So where you're seeing the concentration, and then you have the like the amount bound. Note that the amount bound, like how it's actually reported, is going to vary from method to method. So in the terms of SBR, you're going to get these like relative units. And so basically what you'll get is you'll get something, if you measure it out like this, we have a range of concentrations, and you measure their relative units. You'll get a curve like this. And note your axis. So this is a log scale, and that's why we're seeing a sigmoidal curve. Remember that even those non-cooperative binding is going to look sigmoidal on one of these plots. So you'll get some curve like this, which is one of those curves like what we've been used to. But more typically, when you're looking at SBR data, you're actually going to be something like this, where it looks more like these like shark fins. What this is because what it's showing you is the measurements for the measurements at those different concentrations. So here it's showing you basically the fraction bound or the relative units at these different concentrations. And here it's actually showing you the kinetic data for each of those measurements. So you can see that what you have is basically you have the association, you have it reaches equilibrium, and then you have the dissociation. So SBR is able to measure all those different phases and then fit the equilibrium constant based on those association and dissociation constants, as well as being able to do measurements from equilibrium. So you can get all this sorts of different data, and this is the method of SPR. A similar method is Biolayer interferometry, or BLI. It uses the same principle that if things are bound to the surface of some sort of sensor, and then you shine light at that sensor, it's going to change the influence, it's going to change how the light interacts with that surface and how the light's reflected. What's different about biolayer interferometry, or BLI, is that here what you're dealing with is you're dealing with on the surface of a sensor rather than on a chip. So this has some technical advantages, also some disadvantages, but this would be your BLI. Again, it's similar in principle, but it is slightly different technologically. But that is your BLI. You can get that same sort of kinetic data as well as your equilibrium data. A third method that can also give you information about kinetics and stuff is isothermal titration calorimetry, or ITC. Basically, ITC is going to involve like calorimetry, so measuring heat. You're going to have this kind of container that has two cells in it. It has a reference cell and a sample cell. And what it's going to do is the reference cell is going to kind of like be set at some sort of temperature. And then the sample cell, this is where things are going to be happening. You're going to have your binding interaction happening here, and you want to keep it matching the reference cell. But when you have those binding interactions, they're going to change the free energy. They're going to change the heat of that heat in that cell. And this is going to make it so that it's out of balance with that reference cell. The machine can then compensate and see, okay, how much heat do I have to apply or remove in order to get it back to match that reference cell? And this can be measured to help us determine how much of the molecule is actually going to be bound. So ITC is a way that we can then measure this. So again, you're trying to like compensate for the changes in energy that happen from these binding interactions so that you can keep it matching to that reference cell. Another method is Microscale Thermophoresis, or MST. And this method, you have these little capillaries, so these little glass tubes, and then you have this laser. You have your molecule that's going to be labeled with a fluorophore, so with a fluorescent molecule. And then what's going to happen is that this light is going to excite those fluorophores, get them to give off light, but it's also going to heat things up. And when it heats things up, basically those molecules, well, they're going to get energy, they're going to move away, and then they're going to start moving back. Their movement is going to be influenced by how big and bulky they are. It's easier for small things to move more quickly, so they will, but the bigger things are going to move more slowly. And that's by measuring how fast things are moving, you can then measure like how big they are, kind of. So if they're bound to something, they're going to move more slowly than if they weren't bound to something. And this is the basis of MST. Kind of similar in the principle of bigger things moving more slowly is fluorescence polarization, or FP. Also similar to MST, you're going to have your molecule be labeled with some sort of fluorophore. And so remember, a fluorophore is just a molecule that absorbs light at one wavelength and releases a different wavelength. So you have an absorption at one wavelength and an emission at another wavelength. Different fluorophores are going to have different absorption and emission spectra because this corresponds to how much energy it takes to excite one of their electrons, which is going to depend on the chemical makeup of the molecule. Typically, they're going to absorb light at a higher wavelength, and then that excites an electron. That electron gets excited, but it can't stay excited forever, so it's going to fall back down. It has to give back the energy, but it's going to lose some of that energy in things like vibrational relaxation, which is non-radiative, so it's not giving off light. And therefore, the light that's emitted is going to be of a lower energy, a longer wavelength. So this is the basic principle of fluorescence, and we can take advantage of it in fluorescence polarization. Basically, polarized light, if we shine it at a molecule that's moving around really rapidly, the rapid movement of that molecule is kind of going to cancel out all that polarization of the light. And therefore, the emitted light from those fluorophores is going to have low polarization. So you start with polarized light. Then the molecules, it's going to excite those fluorophores, but the fluorophores, because they're in all sorts of different orientations, they're going to kind of cancel out the polarization. So your emitted light is going to be depolarized. But if that fluorescence molecule is, that fluorescently tagged molecule is bound to something big, well, now rotation is going to be slowed. When you shine that polarized excitation light at them, now the molecules are all sort of going to be in the same orientation because they can't rotate as quickly, and therefore the light that you're going to get out is going to be polarized. And so then we get this FP signal, this fluorescence polarization signal. So a couple of the disadvantages of fluorescence polarization and MST are that you have to have that sort of label. But when you have one of these labels, you can have high sensitivity. And so you often don't need as much as you would need for something where you don't have any label. Speaking of labels, sometimes what we use to label molecules is actually radioactivity. So if we're dealing with things like RNA and DNA, especially if we need to have really low concentrations of them, because remember for our binding assays, we want to have a really low concentration of one of the partners so we don't have to worry about depletion of the other partner. One of the ways that we can have a low concentration of something but still have enough signal to see is by using radioactivity. So we can say radioactively label the ends of DNA or RNA using radioactive phosphorus. And this is what I did a lot in grad school, was radio labeling RNA so that I could track it. More specifically, I want to track its binding to a protein I was studying, and I did so using a method called slot plotting. In this method, you basically, it's also like it's a filter binding technique. So I have these kind of like sandwich of filters membranes. And so on the top, I have a nitrocellulose membrane that'll bind protein, but it's going to let through unbound RNA. And underneath that is a nylon membrane that will bind to the RNA. If I then use this vacuum apparatus to flow the liquid through, the protein and any bound RNA is going to get stuck on the top membrane, and the free RNA is going to flow through. And therefore, I can measure the relative amounts of bound and unbound for the two, compare that to the concentration of the protein I had in there, and this is going to enable me to determine the KD. So I spent way too much time in the hot room, which is the room that what we call the room, like hot is kind of like slang for radioactive. So we have like a room that we would do our radioactivity work in. That was our hot room. And I spent so much time in there during during grad school. That was like my home away from home. So the lab was my home away from home, and the hot room was my home away from home. Anyway, another method we can use to study the binding of DNA or RNA to proteins is an EMSA, or an electrophoretic mobility shift assay. Sometimes this is called like a gel shift assay. We're going to talk a lot about this method called SDS page electrophoresis in this class, and we're actually going to be doing it in the lab. It's one of these really core biochemistry techniques where you have this thin vertical slab of gel, which serves as this kind of like mesh. We're then going to take a solution containing unfolded, or what we call denatured proteins, and we use electricity to power it to swim through the gel. The bigger things are going to get tangled up more as they travel through, and so they're going to get slowed down more. And therefore, when you turn off the electricity and get them to stop swimming, they're going to be higher up in the gel. And therefore, we can kind of get an idea about the relative number and size of protein products in the solution. Things are going to look a lot bigger than they actually are if they're bound to one another. And so if we have a DNA or RNA running alone, it's going to travel pretty quickly. But if it's bound to a big old protein, it's going to travel more slowly. And therefore, you're going to get a shift upward in your gel. And you can use this to measure binding affinity. Another acronym starting with an E is an ELISA, or an enzyme-linked immunosorbent assay. We're going to talk a lot about in the lab about how we can like use antibodies to our advantage in biochemistry. So you probably think about antibodies more in terms of your immune system. But basically antibodies are really useful in the lab as well because they're able to detect specific things. So we'll get much more into the details, but basically antibodies have a generic part that's generic for the animal that made them, and a unique part that's going to recognize some specific feature. And we can use antibodies that recognize specific parts of molecules that we're looking for in order to identify those molecules. One way that we can take advantage of this is with an ELISA. where basically what we have is we have a wells of a plate are going to be coated with an antigen. So this is going to be something that an antibody recognizes. Then what we're going to do is we can flow antibodies over a sample containing antibodies over the sample. If the antibody recognizes the antigen, it'll bind, and then we can then detect that bound antibody. There are also other methods where basically what we do is we have the antibody bound to the chip, and then we flow something with antigen over it. So remember, antigen is going to be the thing that the antibody binds to. And this way we can detect that bound thing, detect that binding partner binding to the antibody. We call it an ELISA or enzyme-linked immunosorbent assay because often that antibody that we're using, one of the antibodies that we're using is going to be attached, or again, we can call it conjugated to something, to an enzyme that's going to allow us to detect it, such as HRP, which is going to convert a chemical into a product that we can see. So again, don't worry about all these details, but basically know that an ELISA is going to be a method that we can use to, in a plate, to measure binding of two molecules, taking advantage of the properties of antibodies to recognize specific molecules. So those are some of the main methods that we use, but there's a lot more methods too. Some of these include things like FRET and BRET. So we talked a little bit about fluorescence. So there's also this thing called FRET, or Forster resonance energy transfer. You might also see it called fluorescence resonance energy transfer. The basic idea is that, remember, with fluorescence, we have light of one wavelength being absorbed and then emitted at a different wavelength. What can happen is that there can be a molecule nearby that absorbs that second wavelength. It can either then just kind of quench that, and so that it doesn't give off light afterwards, and this would be like quench fluorescence, or it can absorb that new light and then give off light of a different wavelength. We can call molecules that absorb light chromophores, or the parts of the molecules that absorb light chromophores. So if we have one chromophore that absorbs light at one wavelength and gives off light at another wavelength, and then we have another chromophore nearby. If that chromophore recognizes or absorbs that wavelength that the first thing gave off, well, now there's a couple things that could happen. It could either absorb it and then kind of just like fizzle it off as heat and stuff, in which case what you would get was you would have some sort of like quench fluorescence. Or what can happen is maybe it absorbs that and then it emits a different color light. So it's going to give you a different color emission if you have the chromophores near one another than if they were far apart. So basically the light that you see is going to not be that light that's given off by the first thing, but instead the light given off by the second thing, or the absence of light. And so that light from the first thing isn't actually given off radiatively. Instead, it's kind of transferred through this FRET process. This can only happen though if the things are close together. And so by measuring the light given off or not given off, you can then measure if these molecules are in contact with one another, or at least close together. There's something similar called BRET. And so basically what's going to happen is that it's going to be based on a chemical luminescence reaction. So luminescence is just the giving off of light. If the excitation that gives way to that light is from light, then we call it fluorescence. But if it's from some other reaction, we can just refer to it as luminescence. And if it comes from a biological reaction, we often call it bioluminescence. So with BRET, we'll have a bioluminescent detection method, and it's using that luciferase. So it's one of these enzymes that's going to be able to convert a chemical into a light that we can see, or some sort of color, or things like this. So same principle with BRET, except that here we're dealing with luciferase typically, instead of dealing with something like one of those conventional fluorophores. And this can have things like less background signal and things like this, so there are certain advantages. In addition to those sort of like quantitative experiments, we're actually getting numbers. There are more like qualitative experiments, so we can kind of just see like, do these things bind? And remember that binding is all relative. So whenever we're trying to see if things bind, we need to be very, very aware of what concentrations we're using, aware of the salt concentration. Remember, the higher the salt concentration, the harder it is for things to bind one another based on those charge-based interactions, because the salt's going to be kind of competing with it. So if you really want to see if things are bound tightly, you often use a high salt conditions. And remember that if you have a ton of stuff around, well, then it's likely to be bound, even if it's not likely to be bound at physiological concentrations, which is why it's really important to know the KD and things like this. But if you just take a sample and you want to see, okay, what's bound to it, there are methods that you can use, such as pull downs. And one of the main types of pull downs is a co-immunoprecipitation or co-IP. Remember, our antibodies can recognize specific features. And so we can have an antibody that recognizes a specific feature on a protein. Often this is going to be one of those like tags that we put on it. So commonly these are going to be things like flag tags or his tags, strep tags, various things like this. We have antibodies that can recognize those, and we have these antibodies that are conjugated or attached to like magnetic beads often. We can then mix those beads with a mixture containing our bait protein, the one that is going to have that label that the antibody recognizes. And then we can kind of use a magnet to isolate the beads that are bound to it, wash away the stuff that's not bound, and then elute or unbind our the bound protein along with the stuff that's bound to it. And then there are methods that we can use to go and see what's bound. One of these methods is going to be a Western blot, which we'll talk more about, which basically takes one of those SDS pages and then it actually goes and probes to look for if specific proteins are present in that gel. And so a co-IP is one type of a pull down, but we can also use other types of pull downs where instead of using antibodies, we use things like the kind of resin that we use when we're doing purification. Speaking of purification, remember we have that size exclusion chromatography that's going to allow us to separate proteins by size. We're often using it in a preparative way. So when we're purifying proteins, either just to make them purer, so we can work with them, or we can maybe get their structures. But sometimes we want to use this like a smaller form of chromatography just to kind of get a look at how big the proteins are and whether they're interacting. It's a similar principle to an EMSA, where if two things are bound to one another, they're going to move as if they were a bigger thing. And therefore, if we're separating proteins and complexes based on their sizes, if proteins are bound to one another, they're going to travel as if they were bigger. And we can detect this using analytical size exclusion chromatography. So we call it analytical. We're just using smaller amounts to kind of get a look, rather than preparative, where we're using larger amounts and we're actually trying to purify it for use. If you want to know more, there's this great article, A Guide to Simple and Informative Binding Assays. And so I will post a link to that. And if you want a lot more detail, this eLife article on how to measure and evaluate binding affinities from the Hirschlag lab is incredibly useful, but this is going to be one of those more technical ones. So again, don't worry about memorizing all of these. It is good to have idea of a couple of techniques that you could use. The techniques that you would actually choose if you were doing this in the lab would be based on various things like what equipment you have access to, how much sample you have access to, what type of labels you can use, all these various technical factors that you don't need to worry about. But it is good to just have a general idea of the type of techniques that are out there, maybe be able to name a couple, but not necessarily know when you would use one versus the other. So I hope this helps you approach your papers and not be scared off by these big terms. They're all kind of doing fairly similar things. That is, we're measuring binding affinity and/or kinetics.